Okay, today, first we'll introduce diagnosis and prevention of bacterial infection. This is gonna be the last chapter on general knowledge of bacteria. And after that, we'll introduce the specific pathogens one by one. And we'll introduce cockers first. Okay, quiet. All right. Diagnosis and prevention of bacterial infection. So assume that in several years, you will be a doctor working in a hospital or your own clinic. You have a patient coming into your office, and the patient is having fever, and you got the first impression that this patient might suffering from some bacterial infection. Then what we are learning today is help you to understand, to find out and determine what type of infection it is. Is it a bacterial infection? And if it is, you also want to understand what type of bacteria it is and what type of antibiotic the bacteria is sensitive to. You want to pick the proper medicine for your patient, okay? And also we want to understand, we want to learn how to prevent bacterial infection. First, we want to learn how to diagnose the bacterial infection. What happened? Never mind. So here's the laboratory procedure. It's the standard procedure we can follow step by step to diagnose the type of bacteria. First, once you notice it's possible of bacterial infection, you want to collect the specimen, okay, the proper specimen from the infected site. And after that, from the specimen, you want to examine the morphology of the bacteria. You want to have a quick examination with a light microscope and see what they look like. And also you can check whether the agent or the bacteria is motile or not. So you want to check the live bacteria in the specimen. And also the golden standard till now, we want to isolate and culture the bacteria from the specimen. So that's the isolation and also identification. Once you get a pure culture, you can further study their characteristics, such as their biochemical activity and also other information from the pure culture. And we can examine the antigen from the agent with the immunoassay. So that's, you use the known antibody to detect the unknown antigen from the agent to help you to decide whether the agent or your specimen contains some specific antigen. And we can also detect and amplify the specific DNA, nucleic acid from the patient specimen. And this method is being used more and more in recent years because of the development of molecular biology methods. We can detect the presence of certain specific gene. And in recent years, we can also sequence the nucleic acid, okay, so that we can get detailed information. And also, we can detect the presence of antibody in the patient's serum. If the patient's serum contains certain antibody after infection, especially systemic infection. And the presence of certain antibody can help you to get useful information for further treatment. Something wrong with this computer. I don't have any problem on my computer in this slide. Okay. But once we get a specimen, there are several points we want to pay attention to. The first one is that the quantity of the material must be adequate. So if you collect a specimen, you want to collect enough material for further analysis. And second, the sample should be representative of the infectious process. So you want to make sure that you collect the specimen at the proper time point. If you notice that your patient is already recovered or is almost recovered, then if you collect the specimen, probably it's not representative. You cannot identify the proper bacteria in the specimen. And you want to try your best to avoid any possible contamination. Try to avoid any possible contamination, such as the tools you use, the material you use should be sterile. You want to use the sterile tube, sterile cotton swab, and also needle. Anything you use should be sterile to make sure that you don't induce or introduce any contamination while you collect the specimen. And the next, once the specimen is collected, you want to take the specimen to the lab for further analysis immediately or as soon as possible. We say that promptly. Because you don't want to leave the specimen on your desk for a couple of days. Because usually the bacteria will die. And some bacteria, they are able to produce some autolysin. They can lyse themselves. So after some time, you won't find any live bacteria in the specimen. So you want to send it to the lab as soon as possible. I have a question for you. Once the specimen is collected, is it necessary to put it on ice for all specimens? Do you have to put all the specimens on ice? The answer is no. Usually, well, for a lot of specimens, to keep it at low temperature is good. But that can inhibit the bacterial growth and inhibit the contamination and can protect the cell. But some pathogens, they don't like the low temperature. If you keep it on ice, the bacteria will die and lyse themselves. So you don't want to put all the specimen on ice. So we should follow the file, the standard, to treat any specific specimen. And also, usually we should collect the specimen before the patient use any antimicrobial drugs or antibiotics. So you want to make sure that you don't want to collect the specimen after you subscribe antibiotic to your patient, okay? But usually it depends on the information you have. You have to ask your patient before they come to your office. They might have already used some antibiotics. If they have some antibiotic at home, they might have using the antibiotics. So you need to get that information collected, because that way probably the bacteria you collected might be already killed or inhibited. It's not typical structure, so it's possible. So that's a specimen. And once you get a specimen, and sometimes as a doctor, you can also examine the morphology of the bacteria. Or if you work in the lab, you want to examine the bacteria in the specimen. You can examine both the live bacteria or the stand bacteria. If we want to observe the live bacteria, we usually check their motility to check whether the bacteria are motile or not. The method we use is called ion drop method. So that's a dropout bacteria suspension, which is placed between the cover slip and the glass slide. And we can examine it with a light microscope. It's live bacteria suspension. If the bacteria is motile, you can see that swimming bacteria in the suspension. Okay, let me show you this video on the ion drop method. It's a very simple method to be carried out, but we don't have this experiment for you in the lab. Show you. This is the material required for this experiment. You prepare the glass slide. Then you prepare the cover slip, the small one cover slip. We used this before. Okay. And you have this bacterial suspension. This is pure culture. You put a drop of bacterial suspension at the center of the cover slip. Okay. And in that drop, it contains a lot of bacteria. Okay. Then you apply a little bit of paraffin wax on the glass slide. The paraffin wax is mineral oil, which is very sticky and hydrophobic. Put it on the glass slide. It can make some distance between the cover slip and the slide. Okay. Then you use the cover slip to stick to the glass slide to stick to the cover slip. Then invert. So now, okay, then invert. Now you have the drop hanging from the cover slip. Okay. Then you examine the live bacteria with a microscope. Okay, this is the way how we use a microscope to focus. Okay, okay. This is a very old style microscope. Has anyone used this one? With a mirror to reflect light? No. Our microscope is a little better than this. We have a light source directly installed in the microscope. Now you'll find the drop, the edge of the drop. Okay. This is a low power lens. Then you change the high power lens and adjust to focus. You will see the bacterial cell like this. At the edge of the drop, okay, this is the edge of the drop. At high power of lens, you can see the bacteria moving around. Okay, some of them moving fast. So that's a motile bacteria. If a bacterium is motile like this, what information you can get? They are motile. They should have some special structure. The name of that structure? Flagella? Yeah, flagella. They should have the flagella so that they are motile. So this, we can examine the live bacteria, whether they are motile or not. You can also kill the bacteria and stain the bacteria with certain color, certain dye, and that can help you to examine their morphology in detail. So we can stain the bacteria with simple stain. Simple stain is that you stain the bacteria with one single dye, such as the methylene blue. This is the methylene blue stain. All the bacteria will be stained in blue color. Then you can check it with a microscope. Or you can also run the complex stain, like the gram stain. This is the second lab you will do, the gram stain. So this is the method we can differentiate gram-positive bacteria and gram-negative bacteria. We introduced that before. And you will also do another staining, let's see, acid-fast staining, which is a special method to stain the Mycobacterium tuberculosis. The material we use is a BCG vaccine. Okay. The staining method, the gram staining you will do for the second lab is this procedure. So what we do is that we have the bacterial suspension in the sample. We want to make a smear or a thin smear on the glass slide. Okay, you make a smear with a loop, then you air dry the smear and fix it by heating the slide gently. After fixation, we stain the bacteria with crystal violet first, which is the purple dye. It can stain both gram-positive and gram-negative bacteria with the purple color. Then we treat it with iodine. Iodine will bind to the crystal violet, which can make it harder to be washed away. Then we decolorize. This is a very critical step. Decolorization is critical. We treat it with ethanol and acid. And ethanol can wash away the crystal violet in gram-negative bacteria. But the gram-positive bacteria is more resistant to the decolorization. So after that, the gram-negative bacteria is colorless, while gram-positive still carry the purple color. The final step is a counterstain using a red or pink dye called safranin. Okay, we counterstain with safranin so that after the final staining, the gram-negative bacteria will show this red or pink color, while gram-positive bacteria will show still the blue-purple color, so that you can tell the difference between gram-positive bacteria and gram-negative bacteria. And this gram staining has been invented over 150 or 160 years, but still it's being used in hospitals, because after determining whether it's gram-positive or gram-negative, it can help the doctor to use the proper antibiotic, because usually we use different antibiotic for gram-positive or gram-negative. Okay, this is the second experiment you are going to do. The golden standard for bacterial diagnosis right now in the hospital is still isolation and cultivation. You have to isolate and purify the pathogen so that you can further identify the bacteria. So we can use different culture media to help you to culture the pathogen. There are so many different type of media in the hospital, and such as the most used one is the blood agar, which contains the intact red blood cells, 5% of sheep red blood, or you can use chocolate agar. Chocolate agar look like exactly like chocolate. It's misleading. It does not contain any chocolate at all. It contains the heated blood. So you heat the blood, dry the blood, the color will be like dark brown color. Then you make the dry blood soluble again. it will show the brown color, which look like chocolate. And it is also popularly used in the hospital to culture human pathogens. And there are also some select medium, such as the McConkey medium we introduced before. On the McConkey medium, only the gram-negative bacteria can grow, while the gram-positive bacteria cannot grow. So that we can use McConkey medium to culture the gram-negative bacteria from the human intestine, such as the Enterobacteriaceae families. And there are also anaerobic medium, which is used to culture the anaerobic bacteria. While we culture and isolate the pathogen, we can examine the colony morphology. You check the morphology and check their size, their shape, their color, and their texture. All this information are useful. You want to collect all the information as much as possible, put them together to help you determine the type of bacteria. So this is the isolation and identification. To further identify the bacteria, we want to collect more information such as the cultural characteristic. For example, whether the bacteria requires unique or specific nutrients for their growth. And the famous pathogens, some of them require special nutrients for their growth. For example, the one I used to work on, Legionella. Legionella is a bacteria which can cause pneumonia in our lung. It requires the addition of cysteine, a special amino acid, to grow. If without cysteine, the pathogen cannot grow. After you add cysteine, it can grow. It's possible that this is Legionella. So that's some information. Or the pigment production. Some bacteria produce special pigment, which is very characteristic. You can rely on that information for diagnosis. For example, the Pseudomonas aeruginosa, which produce the water-soluble green pigment. So if you see the whole plate is stained with green pigment, you know that this might be some aeruginosa or some Pseudomonas species. And also we can study their biochemical characteristics to see whether they are able to decompose, which means degrade certain substrate, and also produce certain products like acid or gas. That's also useful information. We can run a serological test to see whether they produce some certain antibody, or you can detect the presence of a certain antigen in the serum, and to see whether they produce certain toxin, and use that toxin to give it to animals to set up animal model. And some toxin might have a specific characteristic so that the animal will show some symptom. They get sick, they get some problem. And we can also run the susceptibility test. This is a test that you did already. Remember that? The first experiment, you use the paper disc, which contain antibiotic in it, and you put it on the surface of the agar. And after that, you culture the plate. If the bacteria you plate on the surface is sensitive to the antibiotic, there will be an inhibition zone surrounding the paper disc, so that in some area the bacteria cannot grow surrounding the paper disc. So that's we call the susceptibility test. You can measure the diameter of the inhibition zone and to see whether the bacteria is sensitive to the antibiotic on the paper disc. Okay, all this information you can collect and put them together, and it will help you to decide the type of bacteria. And previously, it relies on people's experience. You have to remember a lot of things to decide the type of bacteria. And nowadays, you can use professional software or even AI to collect this information and put this information to AI. AI can help you to analyze the information and give you some conclusion. And we can also detect the production of specific antigen. You can do this in different ways, such as ELISA or agglutination. We did ELISA before. The one we did is sandwich ELISA. We use ELISA to detect the target protein. The one we did, the target protein is produced by human, but you can also detect some protein produced by some pathogen. So it's the same way, similar way. I don't want to introduce ELISA again. Or we can run the agglutination test. Agglutination test can be carried out in this way. It's called latex agglutination. Latex is a material produced by humans. They make it in latex beads. After that, they coat the beads with known antibody, conjugate the beads with antibody, then they make this bead suspension in a liquid. Then they mix the liquid suspension with antigen. This might be some antigen produced by certain pathogen, or it's possible some antigen from the patient's serum. You mix it together, and the antibody can bind to the antigen, to the epitope, so that after incubation, the antigen molecule would cross-link the beads together. So at the beginning, it's a single bead suspension, but if there is agglutination, the presence of antigen, the agglutination happens so that a lot of beads and molecules will bind to each other to form a big complex. And this big complex will precipitate out so that we can actually see the agglutination with our eyes. So this is the method we use it to detect the presence of certain antigen. Let me show you this video, latex agglutination.
Agglutination assays have been used for decades as a simple method to detect antigenic substances in biological samples. The purpose of this video is to explain how this method works in practice and to expose its limitations. The agglutination assay uses tiny particles, most often latex beads. The beads are coated with a specific antibody against the antigen that you would like to detect. The test is usually performed on a card or glass or plastic slide, often one with a black surface. First, you add a suspension of the coated latex beads to each of the three encircled areas on the slide. Note that the suspension is concentrated enough to produce a milky appearance on the background. Now you add a few drops of the unknown sample that you're interested in testing, but you will also need to use one circled area for a negative control solution that contains no antigen, and another for a positive control solution that contains the antigen of interest. Next, the slide is gently rocked or swirled to mix the beads with the test solutions, and the samples containing the antigen of interest will begin to agglutinate the beads. This will produce the appearance of visible clumps, and the solution itself will turn from milky in appearance to clear and transparent. This transition should occur in the circle with the positive control. If the antigen is present in the unknown sample, then it will form clumps. The negative control circle should remain unclumped and opaque. Recall that the latex beads are coated with a specific antibody so that each bead can bind to numerous antigens. For agglutination to work, the antigen of interest must also be able to bind to multiple beads. Therefore, in this assay, antigens that can be detected are limited to large macromolecules that have repetitive antigenic domains. Molecules like microbial capsules, flagella, or lipopolysaccharides. One long repeating antigen molecule can then attach to several beads, causing them to clump together or agglutinate. So even very tiny quantities of antigens that have lots of repeating antigenic domains can cause visible clumps to form and be detected by this test. This is the basis of the test. Finally, here are some examples of agglutination assays that are used in clinical practice.
Okay, so this is a agglutination test to detect the presence of certain antigen. And we can also run the molecular diagnosis. In biology, whenever we talk about molecular, we are talking about nucleic acid. So this is to detect the presence of specific DNA, such as you can identify the bacteria using its 16S ribosome RNA, which help you to determine the actual genus or sometimes the species of the bacteria. Or we can run a DNA hybridization method or PCR. PCR is widely used in the hospital right now. It can give you results in a couple of hours. And in recent years, because of the sequencing price is getting lower and lower, and doctors would like to do the nucleic acid sequencing, so that can help them to get the information. And DNA microarray. DNA microarray was an kind of old method. It was popular 20 years ago, but nowadays, because of the utilization of sequencing, DNA chip, DNA microarray is not popular anymore because it requires specific equipment and the price is much higher than sequencing. Okay. And serological test, you detect the presence of certain antibody in your patient's serum. So there are several different ways to detect the presence of antibody. You can use ELISA, you can also run agglutination, precipitation, and complement fixation. Yeah, different methods, but I'd like to introduce the complement fixation method. I guess you haven't heard of this method before. So this is the complement fixation test. So what you do is that you collect the patient's serum. Okay, here are two serum, one with antibody in it, one without antibody in it. Then you mix your patient's serum with the antigen. This antigen can bind to this antibody. And also you add in the complement molecules. We learned that in immunology. Okay, you mix them together. If there is antibody in the serum, this antibody can bind to the antigen. Okay, and meanwhile, the complement molecule will be fixed to form this big complex. Okay, so this is the first step. Then you add another component called sheep red blood cell. So that's a red blood cell, and also another antibody which can bind to the red blood cell to mix it together in the same tube. In this tube, the complement molecules has already been used in the first complex, so that even the green antibody can bind to the sheep red blood cell. There is no complement molecule available. It's already been fixed so that the red blood cell cannot be lysed due to hemolysis. So this is the positive test. Positive means that there is presence of certain antibody in the serum. But in the second serum, there is no antibody. And if you add antigen and complement, okay, they will be free in a solution. Once you add the sheep red blood cell and the antibody against the sheep red blood cell, the antibody will bind to the red blood cell and also bind to the complement molecule. And we know that the complement molecule can form the MAC complex on the membrane so that it can lead to the hemolysis to lyse the red blood cell. And finally, this tube will look pink because of lysis of red blood cell. So this is a negative result. Negative means there is no antibody in the serum. So this is a complement fixation test. Okay, so yeah, this is the first topic on first part of the first topic on diagnosis. The rest part of we'll talk about the prevention of bacterial infection. You can use different methods to prevent bacterial infection. And here we'll introduce two major methods. One is the active immunity. So we give the patient, we give the normal person some vaccine or toxoid. We know toxoid, right? Last time we introduced toxoid. The vaccine or toxoid can stimulate our immune system to form immunologic memory so that we are protected. Or you can also use passive immunity. We introduce plasma or globulin, which contains the antibody, into some person's body so that we can provide some urgent, immediate protection. This is passive immunity. This is active immunization. There are different types of vaccines. You can use a live attenuated vaccine. This is the live bacteria, but it cannot cause disease anymore, so that we can introduce this to our body to stimulate the immune system. The example in this group is the BCG vaccine against MTB, against tuberculosis. Or you can use the inactivated, the killed vaccine. So that you culture the bacteria, then kill it with heat or radiation. And this produce the inactivating material, but which can stimulate the immune system to provide protection. The third group is a subunit vaccine. This subunit vaccine, you only use certain antigen, for example, certain protein to stimulate the immune system. And usually it's more safe because it's a pure protein. And in recent years, there is another group of vaccine become mature. That's a messenger RNA vaccine. During the COVID-19 pandemic, there are several messenger RNA vaccines became available, and it can provide people with high efficiency. Has any one of you received messenger RNA vaccine during the pandemic? No. COVID-19 messenger RNA vaccine. Me neither, because in China we don't have it. Well, it's available in Hong Kong, in America, and also in Europe. So, but this vaccine is very efficient. Or you can use toxoid. We introduced this before. So that's a denatured toxin, which can stimulate the immune system, but it's not toxic anymore. This is active immunization. While for passive immunization, that's the artificially acquired immunity through a short-term immunization achieved by the transfer of antibody. So we can use purified antibody or the serum from immunized animal. We can use human immunoglobulin or animal blood plasma containing the antitoxin, the antibody. Okay, so that's a passive immunization. Okay, we'll have a break and come back and talk about herd immunity. Herd immunity. Has anyone heard of this term before? Have you ever heard of it? Yeah. Yeah, we never heard of it before the COVID-19 pandemic. And there was a debate on herd immunity for some time. So herd immunity, first we want to understand what is herd. What is herd? That's a herd, right? And this is also a herd. So herd means population, a group of people, a group of animal, we call a herd. So herd immunity is a form of immunity that occurs when the vaccination of a significant portion of the population can provide a measure of protection for individuals who have not developed immunity. So it's a long sentence, which is not easy to understand. Let me show you how it works. This is herd immunity. Okay, I'll explain this in detail to help you understand. How to set up herd immunity? Let me show you in 3 different groups. In these people, this population, yeah, you. Okay, in this group, the blue persons are not immunized, but still they are healthy. While here the yellow color indicates those people who received the vaccine, who are immunized. The red ones are those ones who didn't receive the vaccine, not immunized. They got sick, they got infected, and they are contagious. Contagious means that they are able to spread the germ. In the first group, you can see that all the people are not immunized, but they are healthy. But here, the 2 infected persons join this group. The 2 red ones join this group. Because all the people are not immunized, they are able to spread the germ to the surrounding people. So finally, in this group, most of the people will get infected. So which means that most of the people in this group are not protected. While in the second group, okay, only a small portion of the whole group are immunized, the yellow ones. And when these 2 persons join this group, they can also spread the germ to the surrounding persons. So finally, again, a majority of the whole population will get infected. So which means that there's no protection. While in the last group, you can see here, a big portion of the whole population have already immunized. They are the yellow ones, okay? Even here, even the 2 persons who are sick join this group, they are able to spread the germ but the people surrounding them are already immunized. They cannot get infected. So it's like there is a barrier surrounding those sick person. So prevent them to, to spread the contagious germ to other people. So finally, the final result is that the majority of the whole population will get protected. Even those blue ones in this population who haven't received the vaccine, they are also protected. And those persons who cannot receive the vaccine might due to different reasons. For example, if they are very old or very young, or they are sick due to certain disease, they cannot receive the vaccine, or the vaccine cannot work in their body. They are also protected in this group because of herd immunity. So this is important to set up the herd immunity to finally end some pandemic. For example, the COVID-19. We suffered for several years, but finally the COVID-19 infection gets lower and lower because people receive the vaccine or they get infection naturally. After they get infection, they are immunized for a short period of time. And before the invention of the vaccine, humans have already suffered from a lot of pandemics in the history. And every pandemic finally went down because of the setup of herd immunity due to natural infection. So it's humans paid a lot of cost because we didn't have vaccine, but still we can go through the infection. So the most recent example is happened 100 years ago, the 1918 flu pandemic. At that time, we didn't have any vaccine available, but finally, the human society survived because after a majority of the whole population got infected, the humans are immunized. They can prevent the spreading of the virus. So this is a herd immunity. It's not mentioned in our textbook. It's not mentioned in immunology textbook, but it's a very important concept for us to understand. So that's why I added it here.